Think about losing your sight, bit by bit, until everything just goes dark. That's the stark reality for a staggering 1.5 million people in the U.S. alone living with dry, age-related macular degeneration, or DMD.
Yeah, it's devastating.
And the options to stop this, frankly, they're pretty thin on the ground.
Very limited, yeah.
What makes it even more urgent is that experts predict the number of people affected could nearly, triple by 2050.
Triple. That's huge.
This isn't just a medical problem. It really feels like a tidal wave on the horizon.
It truly underscores a pressing need, doesn't it? When a condition has such, devastating consequences and our toolkit to fight it is so limited, the quest for innovative solutions becomes absolutely critical.
Exactly. And that's where our deep dive today takes us. We're not lingering on the bleakness, but instead shining a light on a potential, maybe a game changer.
Okay.
How artificial intelligence is entering the world of scientific discovery to accelerate the search for new treatments. Specifically, we're going to unpack a really intriguing AI system called Robin.
Robin. Yes, detailed in that research paper our listeners shared.
That's the one.
So our mission in this deep dive is to really get under the hood of this Robin system.
Right.
We want to understand how it works, its magic, what it has already revealed about DMD, and maybe most excitingly, what this could mean for the future of discovering new medicines, for all sorts of diseases.
Consider this your backstage pass to the future of the lab. As we mentioned, our main source is the research paper itself, which really delves into how this ingenious system was built and, what it can do.
Okay,
let's jump in and meet Robin.
All right, so Robin is described as a multi-agent system.
Okay.
And what's fascinating is its ambition to essentially automate the intellectual heavy lifting of scientific discovery.
Heavy lifting, yes.
It's not just about number crunching, it's about trying to replicate the thinking process a scientist goes through.
And it sounds like it's the first system to really put all the pieces together in one go.
Pretty much, yeah.
We're talking about sifting through mountains of existing research, coming up with new ideas about how diseases develop, suggesting ways to test those ideas in the lab, analyzing the results, and then using that information to refine its initial thoughts. It's like the entire scientific method, but, powered by AI.
That's a significant leap forward. I mean, traditionally, these steps are handled by different specialists or teams, which takes a lot of time and coordination.
Right.
Robin brings these crucial functions together through specialized agents. We have Crow and Falcon, which are like incredibly powerful research assistants.
Okay, Crow and Falcon.
Crow can quickly summarize key findings, while Falcon does let's say, the deep dives into scientific papers, clinical trial data, and even databases like the Open Targets platform.
Okay, so Crow gives you the rapid overview, the quick look.
Exactly.
And Falcon is your tireless investigator, sort of digging into all the details.
Yeah.
Makes sense.
Yeah.
And then there's Finch. What's Finch's role in this AI dream team?
Finch. Finch is the data analysis whiz.
The whiz, okay.
It's equipped to handle complex experimental data, things like RNA sequencing data. Think of it as a way to see all the instructions being read inside a cell at once.
Right, like seeing all the genetic instructions in action.
Precisely. And flow cytometry results, which help us sort and analyze cells based on specific characteristics. Finch is like your super skilled biostatistician, but in AI form, ready to make sense of complex biological data sets.
Okay, so we've got the research powerhouses Crow and Falcon, and the data detective, Finch. How do they all, collaborate in this ongoing cycle of discovery?
It's a continuous feedback loop, really.
A loop? Okay.
Robin starts by generating initial ideas, hypotheses based on what Crow and Falcon find in the existing research.
Right.
Then it proposes specific experiments to test these ideas in the lab. Once the experiments are done and we have data, Finch steps in to analyze it.
Makes sense.
The results from Finch then feed back into Robin, allowing it to refine its initial hypotheses and suggest the next set of experiments. It's this constant learning and adaptation based on real-world data that makes it so powerful.
Wow. It's like having a relentlessly curious scientific partner that never gets tired and is always learning.
Kind of, yeah.
Now, the research paper really focuses on how Robin was used to tackle DMD. Considering how common and, devastating this condition is, it seems like a really important test case.
Absolutely. As we discussed at the start, DMD is a leading cause of irreversible vision loss, and our treatment options are just so limited. So the potential to discover new therapies is enormous. The researchers essentially set Robin the challenge, find new ways to treat this condition.
All right, so how did Robin even begin to wrap its digital mind around DMD? What were its initial lines of thinking?
Well, Robin started by poring over a vast amount of scientific literature. I think it was around 151 research papers specifically related to DMD.
151 papers.
Okay. From this huge pool of knowledge, it identified 10 biologically plausible ways that the disease might be progressing. 10 potential mechanisms.
10 potential culprits right off the bat. That sounds like a pretty efficient start.
It really is.
How did Robin then narrow down which of those to focus on?
Well, the system didn't just list these 10 mechanisms, it actually ranked them based on how relevant they seemed to the disease and, crucially, how likely it would be to find a treatment targeting them.
So targetability was key.
Exactly. And interestingly, Robin identified boosting the waste-clearing ability of retinal pigment epithelium, or RPE, cells as a particularly promising therapeutic strategy.
RPE cells. Okay, those are the support cells in our retinas that are crucial for healthy vision, right?
Yeah, it's them.
And this phagocytosis is basically their way of, like, taking out the trash, clearing debris.
Precisely. It's their cellular cleaning system. And the idea is that in DMD, this cleaning system might not be working properly. It gets overwhelmed or slows down.
I see. So the trash piles up.
In a manner of speaking, yes. And Robin didn't just flag this up as important. It went further and suggested a specific type of lab test, a flow cytometry assay, to see if certain drugs could actually improve this waste-clearing function.
Wow.
Using either RPE cells taken from patients or a commonly used lab-grown RPE cell line.
That's incredibly specific. So Robin not only came up with a potential disease mechanism to target, but also provided a concrete way to test drugs against it.
Exactly. The whole package.
What happened after that? Did it start searching for potential drugs?
Yes, it did. Robin then reviewed an even larger collection of research, maybe around 400 papers this time.
400 more. Okay.
Specifically looking at studies on RPE waste clearance, phagocytosis, and existing drugs that might influence it, as well as the broader field of DMD treatments. From all this, it proposed 30 existing drugs that could potentially enhance this cellular cleaning process.
30 drugs that are already available. Wow. That sounds like a fantastic shortcut in the often long and expensive journey of drug discovery.
It really does. Finding a new use for something we already know is relatively safe. That's drug repurposing, and it's a huge advantage.
Definitely.
And to evaluate these 30 candidates, Robin used Falcon, the deep dive agent, to generate detailed reports on each one. These reports explained why each drug might be effective, based on the literature, and also pointed out any potential drawbacks or unknowns.
So a pros and cons list for each drug, basically.
Kind of, yeah, but very detailed and evidence-based. These reports were then put through a sort of AI-powered competition, an LLM-judged tournament, they called.
A tournament, like pitting the drug reports against each other.
Exactly. Ranking them based on the strength of the scientific reasoning, their known properties, safety profiles, and the quality of the research supporting them.
An AI drug face-off.
Yeah.
That's a really innovative way to prioritize potential treatments. So which drugs emerged as the frontrunners from this tournament?
The top 5 candidates were selected for the first round of laboratory testing.
Okay, the top 5.
These included compounds like exendin-4 fingolimod and one called Y27632, which is a type of drug known as a ROCK inhibitor.
Y27632, a ROCK inhibitor.
Okay. These were then tested in that waste clearance assay that Robin had suggested, using special fluorescent beads that light up when they're engulfed by the RPE cells.
Right, so you can measure how much trash the cells are eating.
Exactly. And Finch, the data analysis expert, jumped back into the picture at this stage, right?
Yeah, Finch's turn.
Absolutely. Once the lab generated the data from these experiments, it was fed into Finch. Finch autonomously analyzed this data, quantifying exactly how much each of the top 5 drug candidates improved the RPE cell's ability to engulf those fluorescent beads.
And what did Finch find?
Interestingly, the analysis showed that Y27632, the ROCK inhibitor, did indeed boost waste clearance significantly, which supported Robin's initial reasoning based on the scientific literature it had reviewed.
So the AI's prediction held up in the real world. In the lab dish, anyway.
It did. That's a significant validation.
But the story doesn't end there, does it? Because Robin then suggested a follow-up experiment to dig even deeper.
That's right. Based on those initial encouraging results with Y27632, Robin recommended performing RNA sequencing on the RPE cells that had been treated with it.
Okay, back to the gene expression level.
Exactly. The goal here was to get a much more detailed understanding of the changes happening inside the cells at a molecular level specifically. Which genes were becoming more or less active when this drug improved waste clearance?
And Finch was back on the case to make sense of this new layer of information.
Indeed. Finch performed what's called differential gene expression analysis. Think of it as comparing the entire library of active genes in the treated cells versus the untreated cells to see which ones were significantly different.
Finding the key changes.
Yes, and this analysis revealed that Y27632 wasn't just directly affecting the waste disposal machinery, it was also triggering changes in the activity of genes involved in other important cellular processes.
Like
the cell's internal scaffolding, the actin cytoskeleton also cell signaling pathways involving small GTpases, and even another cellular cleaning mechanism called autophagy.
That's fascinating. So a single drug having potentially multiple beneficial effects that all contribute to the desired outcome, cleaning things up in different ways.
It seems that way, and the paper highlighted one specific gene that was significantly more active.
Yes, ABCA1. Finch's analysis showed a remarkable threefold increase in the activity of this gene in the cells treated with Y27632.
Threefold, okay. And why is ABCA1 interesting?
What makes this particularly interesting is that ABCA1 plays a crucial role in removing fats or lipids from cells.
Ah, lipids. That's relevant to DAMD, isn't it, with the drusen deposit?
Exactly. Those deposits under the retina are rich in lipids. Plus, ABCA1 belongs to the same family of transporters as another gene, ABCA4, which has already been strongly linked to the development of macular degeneration.
Okay, so there's a known connection there.
And furthermore, ApoE, a protein that works with ABCA1 to transport fats, has also been identified as a potential therapeutic target for DMD, so it all starts to link together.
Okay, so Robin not only found a drug that works in the assay, but also potentially uncovered a new key player, or at least highlighted a known player, ABCA1, in the context of this drug's effect on DMD pathology. That sounds like a major discovery in itself.
It feels like it. And this then led to another round of investigation, didn't it? Another iteration.
Exactly. Building on these new insights from the Y27632 experiments and the RNA-seq, Robin proposed another set of drug candidates to test, refining its search.
Smarter search this time.
Hopefully. This time, 10 drugs were selected for laboratory testing, and once again, Finch analyzed the resulting data.
And the big reveal from round 2.
This second round of experiments revealed that ripasudil, another ROCK inhibitor,
Another one, okay.
that is actually already approved for treating glaucoma in Japan.
Oh, wow, already in use.
Yes, already in use for another eye condition. Ripasudil was even more effective than Y27632 at boosting RPE cell waste clearance.
How much more effective?
It showed a remarkable 7.5-fold increase compared to the untreated cells.
7.5 times. That's huge compared to the first round.
A very significant improvement, yes.
Wow. So Robin essentially took the initial promising finding with Y27632, and then through another cycle of reviewing research, analyzing data, generating hypotheses, identified an even better drug, ripasudil,
Yes.
and one that's already being used in patients for something else. That sounds like a massive leap forward for potential DMD treatment.
It really is. And what's particularly significant, the paper points out, is that while ROCK inhibitors like ripasudil had been suggested before for wet AMD,
Right, the other type with blood vessel growth.
the suggestions were mostly related to stopping that abnormal blood vessel growth. Robin is the first to propose them for dry AMD, specifically based on their effect on this crucial waste clearance process, the phagocytosis.
Ah, so a completely different rationale for using the same class of drugs.
Exactly. This really showcases the AI's ability to connect existing knowledge in completely new and insightful ways, finding novel mechanisms for known drugs.
So let's just recap for a second. Robin tackled this massive challenge of finding new ways to treat DMD.
Yep.
And through its automated process of integrating research and experimental data, it successfully identified ripasudil as a really promising candidate.
Seems so.
And this isn't just a lucky guess. It's based on a biologically relevant mechanism, that waste clearance, that the AI itself identified and helped validate through suggesting the right experiments.
Precisely. Robin demonstrated its power to synthesize a vast amount of existing knowledge, generate novel and well-supported hypotheses, and pinpoint existing drugs that could be repurposed.
drugs that already have a known safety profile because they're used to treat other conditions.
Right, which, as we said, could dramatically speed up the timeline for getting new treatments to the people who need them. Less safety testing needed up front.
It really highlights the potential for AI to break through some of the log jams we often see in drug development, where the rate of new FDA approvals has been a bit of a concern.
Yeah, drug development is notoriously slow and expensive.
And the fact that Robin also generated potential ideas for treating 10 other diseases, as mentioned in the supplementary materials of the paper, things like, I think, certain cancers and neurodegenerative diseases were mentioned.
Yes, there was a range listed.
It hints at just how versatile this approach could be.
That's right. While the DMD work is a powerful demonstration, the system's ability to generate hypotheses across various medical fields suggests a truly adaptable tool for scientific exploration.
But it's not perfect yet, right?
No, definitely not. The researchers themselves point out that Robin is still evolving. There are areas for further development,
Like what?
like refining its ability to generate very precise experimental instructions, executable protocols, and improving how Finch autonomously generates its data analysis requests or prompts.
Making it even more independent.
Exactly. And also, more work is needed to ensure that AI's judgment in evaluating hypotheses aligns even more closely with the nuanced perspectives and sort of the intuition of human scientists.
Right. The human touch is still important.
For now, yes. But it definitely feels like we're on the verge of a new era in how scientific discoveries are made.
So what are the key things you, the listener, should really take away from this deep dive into Robin?
Well, I think the core message is that Robin, this innovative AI system, successfully identified a promising new treatment avenue and a specific drug, ripasudil, for dry, age-related macular degeneration.
Yeah.
And it achieved this by autonomously connecting a huge amount of scientific literature with experimental results. It really underscores the transformative potential of AI in accelerating the entire scientific discovery process.
And the real aha moment for me, I think, is seeing how Robin made connections that maybe human scientists hadn't explicitly made before. Linking the critical role of waste clearance in RPE cells with the ability of ROCK inhibitors like ripasudil to enhance that specific process for this disease. It's like the AI provided a kind of shortcut to a potentially major scientific breakthrough.
Absolutely. A way to see patterns across vast data sets that might be missed otherwise. And this leads to a really important question for you to think about. If AI systems like Robin become increasingly central to scientific research,
Which seems likely.
how might this fundamentally change our understanding of diseases and the way we develop new treatments across a vast range of medical challenges? It's a future where the speed of discovery could be dramatically increased, and the possibilities are, well, truly exciting to consider.